Today we will be covering concept 8.1, genes are particulate and are inherited according to Mendel's laws, pages 150 through 158 from Principles of Life, second edition for the AP course, Hillis et al. Now, in this lesson, we're going to be going through the basic principles that Gregor Mendel discovered. Breeding of plants and animals had yielded two hypotheses by the 19th century, either bleeding inheritance or particulate inheritance. So, with blending inheritance, gametes contain determinants, what we call genes, that blended when gametes fused during fertilizations. Particulate inheritance, on the other hand, each determinant was physically distinct and remained intact during fertilization. So by the 1860s, Gregor Mendel used a scientific method in studies of his garden peas, and that really supported the particulate hypothesis. Garden peas were easy to grow, manipulate, created this model system that was easy to replicate and produce large quantities of offspring. So peas will typically normally fertilize, meaning that the male parts, in this case the pollen, will directly fertilize the ovule on the same flower, but we can remove the male parts to prevent fertilization, self-fertilization within the same flower. So these characters that Mendel looked at, these are these observable physical features, things like flower color, seed shape, and then we have traits, things like purple flowers, wrinkled seeds, those pairing the forms of each character. So Mendel worked with these true breeding varieties because they were, once they were crossed, their offspring had the same traits as the parents. So if you had pink flowers in the first generation and this pea generation, you would have the same color flowers in the F1 and the F2, so forth and so on. Now, it was when we began to hybridize. This is when we had crosses between organisms with differing characters. So in the first experiment, Mendel crosses plants, differing in just one character, producing monohybrids in the F1 generation. They were then allowed to self-pollinate to form the F2 generation, which created what's called a monohybrid cross. Okay, so figure 8.1 in your text on page 152 gives a summation of how we did this. So in this case, we have green round seeds, green wrinkled seeds. We cross those and we see that all of the offspring in this case seem to be round. So the question is, what happened to the wrinkled seeds? Okay, so because of this, the hypothesis was rejected. There was no irreversible blending of characteristics, and he found that it didn't disappear. This is the F2 seeds. It was that it was covered up by the dominant allele, which we'll get to in a second. So one trait of each pair disappeared in the F1 generation, then reappeared in the F2. This findings led to the rejection of the blending hypothesis because the F1 offspring were not a blend of the parental traits. Only one trait was present, and the trait 2 did not disappear in the F2 generation either. We just saw that wrinkled seed did return in the F2 generation. So all 7 crosses between the varieties that he had gave the same kind of data. Called the trait that appeared in the F1 that was more abundant in the F2 the dominant trait, and the other trait the recessive, and the ratio of dominant to recessive was about 3-to-1. It wasn't precisely 3-to-1, but we're going to say rounding close to 3-to-1. So he proposed that the genetic determinants, these genes, occur in pairs, and they are segregated or separated in gametes. Each plant gets 2 genes from each parent. We now use the term diploid and haploid to describe both of these. Diploid meaning 2 copies, haploid meaning 1 copy. So Mendel concluded that because of this, gametes contain 1 copy of each gene. The resulting zygote has 2 copies. This is right after fertilization, when the whole copy from one parent has come together with the whole copy from a second parent. Different traits arise because there can be different forms of a gene. Now we know this is being called an allele for a particular character. So things like green peas or yellow peas or wrinkled peas or round peas. Okay, so the phenotype, this is this physical appearance of an organism. Okay, the genotype, the actual genetic makeup of it. Again, these capital letters designate the dominant or recessive alleles are noted by lowercase letters. Okay, so round seeds can be the result of 2 different genotypes. So all we have to have is that one dominant allele. Okay, so we can have big R or big R, little r. As long as that one dominant allele is there, the dominant trait will be expressed. So true breeding individuals are going to have two copies of the same allele. They are said to be homozygous for the same. So big R, or little r, little r. Heterozygous will have two different alleles. In this case, we can only have big R, little r. We would write little r, big R the same way. It's referring to the same genotype. Okay, so in figure 8-2 on page 153, okay, so we have a plant that is homozygous for big R is crossed with one that is homozygous for little r. Okay, so we complete our first Punnett square here, and the parental gametes combine to produce F1 plants with the big R, little r genotype and a round seed phenotype. So we see big R, little r in all four. Now, if we take two of these and we cross them, the heterozygous F1 plant produces these haploid gametes and self-pollinates. And then we come up here, and what we see is that different combinations of the alleles from each parent produce two different seed phenotypes in the F2 generation, and then the seed phenotypes appear in a 3-to-1 ratio. So we have 1, 2, 3 round to 1 wrinkle. Okay, so Mendel's first law that he came up with was the law of segregation. It states that two copies of a gene separate when an individual makes gametes. Each gamete receives only that one copy. Okay, so gametes from a big R individual will all be R, little r will all be R, and all of their offspring will be big R, little r. Okay, so in the F2 generation, gametes will be big R or little r. Okay, so we can test this by putting into a Punnett square, as we saw in our figure right here, crossing those, giving us the phenotypes for the genotypes in the F2 generation. Okay, so the Punnett square will predict this 3-to-1 ratio in the F2 generation, and Mendel's experimental values were remarkably close to this for all 7 of the character traits that he compared. Okay, so genes are now known to be relatively short sequences of DNA on the much longer DNA molecules that make up chromosomes. We can picture the different alleles of a gene segregating as chromosomes separate during meiosis 1. Okay, so in figure 8.3 on page 154, we have a demonstration of this. So we have our diploid parent here with our homologous chromosomes. We are in meiotic interphase demonstrated right here. So the site on the chromosome is the locus of the gene, in this case with the alleles of big R and little r for seed shape. So we see big R, little r right here. So before meiosis 1, each of the homologous chromosomes will replicate. So we see we've gone from haploid to diploid here. Okay, and at this point, at the end of meiosis 1, the 2 chromosomes have segregated into daughter cells. Okay, so again, we've gone back to haploid here. We have this dominant allele, recessive allele here. So at the end of meiosis 2, each haploid gamete is going to contain one member of each pair of homologous chromosomes, and thus one allele for each gene. So genes determine these phenotypes mostly by producing proteins with particular functions. So the proteins that are made that give green, or the proteins that are made to have our green beans and peas be green is actually because of a protein. The same for the seed covering, whether it's wrinkled or smooth. So a wrinkled, see right here, the wrinkled pea phenotype is due to a lack of starch branching enzyme 1, SPE1, essential for starch synthesis. Now, we can recall from beginning of the course that starch is going to be this long branching carbohydrate chain that's going to aid in a lot of structure in plants. So Mendel verified his hypothesis by doing test crosses. So what he would do is he would cross F1 with an unknown homozygote, which has a recessive genotype. And for example, little r, if the F1 individual was homozygous, all offspring will show the dominant trait. If the F1 was heterozygous, half of the offspring will have the recessive trait. So Mendel's next experiments tested whether the genes distribute independently or in the offspring. So he crossed peas that differed in 2 characteristics, in seed shape and color. True breeding parents would be homozygous dominant, and it would be homozygous recessive, so round yellow seeds and wrinkled green seeds. So the F1 generation is big R, big Y, little y. These are all round yellow because they have this one dominant allele present in both. So he took these, crossed them all, and this is called a dihybrid cross. And Mendel asked whether in the gametes produced by big R, big Y, little y, the traits would be linked or segregate independently, essentially saying, are the dominant traits going to be linked? Are the recessive traits going to be linked? Is it going to be one dominant and one recessive together every time? That's what he was trying to determine here. So 2 possible outcomes of the dihybrid cross. If the R-linked gametes would be big R, big Y, or little y, they would have 3 times more yellow round than wrinkle. The ratio would be 3-to-1. If independent gametes if independent gametes could be big R, big Y, little y, big R, little y, little r, big Y, and we would have 9 different genotypes, and the 4 phenotypes would be in a ratio of 9-to-3 to 3-to-1. So using figure 8-5 on page 155 in your text, this is what Mendel did. It's demonstrating it right here. So we test round peas of undetermined genotype. Figure 8-5. And we have this homozygous dominant here, homozygous recessive. We cross that to get our 4 gametes right here. Okay, and when the F1 plants self-pollinate, the gametes combine randomly to produce an F2 generation with 4 phenotypes in a 9-to-3 to 3-to-1 ratio. So our 4 phenotypes that we see are yellow round, green round, yellow wrinkled, and green wrinkled. Okay, and we know this today as Mendel's law of independent assortment. Okay, so alleles of different genes do assort independently during gamete formation. It doesn't always apply to genes near each other on the same chromosome, though, but chromosomes do segregate independently. Okay, so it is possible, with genetic recombination and meiosis, that groups of genes can be inherited together just because of their likelihood of being crossed over together. Okay, so figure 8-6 on page 156 shows this, okay, of how meiosis accounts for independent assortment in alleles. So starting up here at the top, we have our 4 different chromosomes here. Okay, the same traits that we looked at, seed color and seed shape. And when homologs line up on either side of the metaphase plate during metaphase 1, big R, little r go. Okay, so we can see that they line up, but it does not determine where Y and Y go. Okay, so we see we have these different locations here. So we don't, have this big R, this big Y right next to each other. They're actually on completely different chromosomes. Okay, so meiosis continues, and we end up with these distinct chromosome pairings here. Okay, the different genotypes that we talked about, the big R, big Y, little y, little r, big Y, and big R, little y, all being represented here. Okay, so one of the reasons that Mendel was successful was his use of large sample sizes. At the beginning of the lesson, I talked about pea plants being a model system. This model system allows for relatively ease of studying a generation. Typically, model systems will have large numbers of offspring as well, and he could count as many progeny as possible. Progeny referring to offspring. So after his work became recognized, geneticists began using probability calculations to predict expected ratios of genotypes and phenotypes and statistics to determine whether actual results match the predictions. And they found that they pretty much did. Okay, so again, he was doing this all in the garden of his monastery in the 1800s. And even today, with the most complex genetic studies, we're still finding that the probability numbers do pretty much stay within the expected ratios that Mendel found over 150 years ago. So when we talk about probability, it just means the likelihood that an event can happen. Okay, so it's probably if it's certain to happen, it's 1. If it can't possibly happen at all, it's 0. And the probability is always somewhere between 0 and 1. Okay, so 2 coin tosses, for example, are independent events. It's a half chance that you'll get heads both times. So we follow the multiplication rule here. So if you're flipping a coin twice, you have to multiply that fraction, 1/2 times 1/2, to give us the probability of flipping heads twice in a row. Okay, so we take this, figure 8-7 on page 157 in your text. So we take this dime and this penny, we flip them over. We've got 1/2 chance of getting heads, 1/2 chance of getting heads again, 1/2 of the penny, same thing there. Okay, so we always have to multiply these because it shows the general likelihood, the probability that we could have these different combinations, heads, tails, tails heads, and tails tails, respectively. So in a monohybrid cross, after self-pollination of an F1, big R, little r, the probability that an F2 generation offspring will have the genotype big R is 1/2 times 1/2, because we have 1/2 of this genotype and 1/2 again being present. Okay, the probability that the offspring will be little r genotype is the same, because we have half likelihood from each parent, each gene being present there. So the probability that offspring will be heterozygous. Okay, so there are two ways to get a heterozygote: big R, little r, or little r, big R. Because of this, we use the addition rule that states that the probability of an event that can occur in two or more different ways is the sum of the individual probabilities of those ways. So instead of multiplying, we now have 1/2 plus 1/2, 1/4 plus 1/4, giving us 1/2. So when we look at this, it gives us this 1:2 to 1 ratio that's there. Now, sometimes we need to have a way to see how Mendel's laws actually work in human generation. So human pedigrees can show Mendel's laws, and humans have very few offspring comparatively to pea plants. Pedigrees do not show the clear proportions that the pea plant showed. It shows oftentimes how a gene can be carried and transmitted in a family lineage. Pedigrees can show patterns and can be used to determine whether a rare allele is dominant or recessive. Okay, so pattern of inheritance: if a rare allele is dominant, every person with the abnormal phenotype is an affected parent, or either all if homozygous parent, or half if heterozygous parent of offspring in an affected family are affected. So let's put this into some picture right here. So page 157, figure 8.8. This is part of the whole pedigree that's shown here. Okay, so over here, these filled-in shapes, these refer to affected individuals. Okay, so every affected individual has at least one affected parent. You can see that commonality here. Okay, we have about half of the offspring of any time that we have an affected parent seem to be affected as well. So we have 1 out of 4 here, 3 out of 5, 2 out of 3. That averages out to 1/2. Okay, so affected people often have two unaffected parents. If in an unaffected family, 1/4 of children of unaffected parents are affected, those parents are heterozygous. That means they carry the trait, but they are not affected by it. To represent this, we use our split boxes like this. So we have individuals who are carriers, but they are not affected. So we see it takes all the way until generation 4 to have an affected individual, and that is because in this case, yikes, these two cousins, they had a baby together. They had offspring, and they have an affected individual. So this is oftentimes why you hear so much about health problems in individuals who are inbred. Okay, so the map of the royal family is a great one to look at to see the carrying and transmission of different traits within a family. And if you look at this closely, and I want you to put in the comments below, where do we see family members coming together, in this case, to produce offspring? Okay, so put it down in the comments below, and you should be able to see there. So if you have any questions, put them in the comments below, or make sure to bring them to class.